Hello and welcome back, students. Today we'll be covering Chapter 8, Microbial Metabolism. The inside of any cell, even tiny prokaryotic microbes, is a complex concert of chemical reactions, thousands per second, all dedicated to keeping the cell alive. And although most microbes are structurally simple, their metabolism is complex. Metabolism is defined as the chemical reactions involved in the buildup and breakdown of nutrients within a cell. Unlike eukaryotes, which have a pretty basic and limited form of metabolism that they can execute, prokaryotes can live on cellulose, petroleum, sulfur, ammonia, and a variety of other sources. And what is metabolism? Metabolism is made up of catabolic and anabolic reactions. It describes the chemical reactions involved in the buildup and breakdown of nutrients within a cell. Catabolism includes breaking down of complex molecules into simpler ones. And when we say complex molecules, we mean that they tend to be made up of more atoms. And this usually releases energy in the process, which is then captured by the cell in some sort of a storage molecule like ATP. Anabolism is the opposite of this. It is the building up of complex molecules from simpler ones. And this process usually consumes energy. So, catabolic reactions break down nutritional molecules and extract their energy in the form of ATP. Anabolic reactions then couple burning of ATP with some other reaction that requires energy. And so this process is a circular process where simple molecules are made into complex molecules with the use of energy, and then complex molecules can be broken down into simple molecules for the production of ATP that can be used for different tasks.
Checkpoint 1. As pictured to the right, the metabolic step of converting one glyceraldehyde 3-phosphate molecule into one pyruvate is an example of catabolism. Is this true or false, and why?
Adenosine triphosphate, or ATP for short, can be thought of as a battery, a rechargeable battery for the cell. In its charged form, adenosine triphosphate is made of three main components: a nucleic acid called adenine, a sugar called ribose, and three phosphate groups, the triphosphate. If one of these phosphates at the very end is broken, it releases a high amount of energy, and therefore it's called a high energy bond. If that bond gets broken, our ATP charged battery becomes an uncharged adenosine diphosphate for two phosphates, and the byproducts are inorganic phosphate, free energy, and our ADP. Of course, when a cell needs to harvest more energy, it can simply recharge that battery by reattaching that inorganic phosphate back to the adenosine diphosphate, but this requires energy. It's very common to see glucose used as an energy substrate. So when glucose is broken down into CO2 and water, it can give off energy that can be captured in order to produce ATP, a charged battery. Then the cell can use that ATP in order to make more complex components. So it might make amino acids, which are the chemical constituents of proteins, and make proteins out of them. This is a form of anabolism, and it requires an energy input. So again, metabolism describes the chemical reactions involved in the buildup and breakdown of nutrients within a cell. Before we can cover metabolism in greater detail, we first need to have a basic understanding on how chemical reactions occur. There's a concept called collision theory that governs when chemical reactions can occur. So as you can see in this example, there are molecules that are crashing together. This occurs within the cell millions of times, and not all of these molecules that bump into each other react with each other. Most of the time they just graze off of each other. In order for them to react, they need to have the right speed when they collide into each other. They need to have the right amount of energy. And if you notice, for this pair of chemicals that react with each other, they have to be in the right orientation. So in these two, they have these little black stems that need to face each other in order for those two to form a bond to each other. So how often do you think all three of those things happen? Particles are colliding randomly all the time, but only rarely do they have enough speed, enough energy, and the proper orientation for them to react with each other. So chemical reactions are actually extremely rare and extremely slow to happen on their own. Consequently, cells evolved a very tricky way to encourage chemical reactions to take place. So, in order to solve this puzzle, the cells evolved enzymes. And enzymes are proteins that catalyze, or in other words, speed up biological reactions by providing better conditions for substrates to react. So again, enzymes are made of proteins, and they can often require cofactors. And cofactors are molecules like zinc and magnesium that help the reaction take place. They could also use coenzymes, which are organic sort of homologs to that, such as vitamins, NAD, etc. The active site of an enzyme is tailor-made for the substrates that can enter into it, and only a very limited range of specific compounds can interact with the active site of any given enzyme, and there are thousands. When all of those three components are together, it's called the holozyme. And because there are thousands of different chemical reactions, you can imagine that there are many, many different types of enzymes that need to be produced. So the ability of enzymes to increase the rate of reactions occurs by lowering what's called the activation energy. In this image to the left, you see, is what's called a reaction coordinate. So on the bottom is kind of a proxy for time. It's the progress of a reaction. And on the y-axis is the amount of free energy. And molecules want to be in a lower state of energy. So think of it as sledding down a hill. The sled wants to be in a lower state of energy, and that's one where it's lower to the ground. So it will slide down to the lowest point in elevation. So if we look at this image, we can see that reactants are kind of in the middle. So they have a lower amount of energy than a transition state does. And the transition state is the state of molecules that's in between the reactants and what happens after the reaction, which is the products. So in order for the reactants to become products, however, they actually have to go uphill a little bit first. So they can't merely slide down the hill into products, or they would have already been products. There wouldn't have been anything stopping them. So this requirement is called the activation energy. It's written E sub A. And enzymes just work by lowering the amount of activation energy that's required. So if you decrease the amount of activation energy, that means that a higher percentage of molecular collisions are going to lead to a chemical reaction. So again, enzymes make reactions faster by lowering their activation energy.
Fill in the blanks. Enzymes make chemical reactions faster by blank their blank blank.
Okay, great. Now that we've seen the theory of enzymes and how they function, let's see how this works in the real world. So, remember how we said that enzymes are tailor-made to specific substrates? Well, here's an example of an enzyme that can react with this disaccharide. And what it's doing is it's cleaving that disaccharide called sucrose. You may have heard of it. It's common just sugar that you buy in the store. And it's adding water to it, which is hydrating the bonds that link those two saccharides or those two sugar molecules together. And when that occurs, it cleaves those two sugar molecules apart. So you go from one disaccharide to two monosaccharides, one sucrose to a glucose and a fructose molecule. So what occurs is those substrates enter the active site of the enzyme. The enzyme transforms the substrates into new products. And then finally, the products are released and the process can repeat. So I want you to take note that the enzyme is never destroyed in this process. So the enzyme is not a product, it's not a reactant. It's just a mediator of this reaction itself. Now, enzymes are very important in biotechnology. Biotechnologists, molecular biologists, often look for ways to produce unique enzymes within a cell biofactory in order to create a certain product. So here's an example, R-Zeta that designs enzymes and then engineers them in a metabolic pathway in order to produce a product that has some sort of economic value to it. And here's an example of them producing levulinic acid, which has a high value and has a high demand in material sciences. Much like cells, and not coincidentally, enzyme activity rates have different optimums. So they have an optimum temperature, an optimum pH, and an optimum substrate concentration. And how efficient an enzyme is at turning over reactions depends on environmental factors. These optimums vary from species to species depending upon their natural environment. For example, enzymes in E. coli, which lives in the human colon, function optimally at human body temperature and approximately a neutral pH. Also, much like cells, this optimum tends to have a slow rise, and then after it's past that optimum for each environmental parameter, it drops off precipitously. And the reason for that is high temperatures can actually damage biomolecules within a cell, and enzymes are one of them. Enzymes are made of proteins, and if those proteins get, too hot or maybe outside of their pH ranges, it can cause that protein to degrade and no longer be functional. So in this example right here, we see this optimum is about 37. So this wouldn't be surprising to find this organism is one that inhabits, some sort of a warm-blooded creature, an animal of some sort, the intestines of an animal likely. Here we can see that pH affects the enzymatic activity, and most enzymes perform the best around a neutral pH. And that's because most internal cells, cytosolic pHs, are close to neutral, maybe a little bit on the acidic side. And finally, is an optimal substrate activity or concentration. So you can imagine if you have so many enzymes floating around, let's say you have 5 enzymes, and they can only react with 5 substrates per minute, then if you double the concentration of substrates, it's not going to be able to produce any more products any faster. So once you reach that, it's called the saturation point, where you'll no longer get any faster enzymatic activity out of it.
Checkpoint 3. What are the 3 conditions that affect the activity of all enzymes?
Humans have known about this for a lot longer than they have known about what exactly enzymes are. And we can see this evidently in the brewing process of beer. So the brewing process starts with the production of malt, which is grain that is germinated and then kiln dried. The germination process allows starches to be liberated, and enzymes are produced in this process. Following the malting, the grains are then mashed, and mashing is when you soak the grains in hot water, and the enzymes become very active in this hot water. And you can see here that there's a variety of different enzymes. Most importantly are the beta amylases and the alpha amylases, which break down starches. And they have their own specific temperature. We can see on the y-axis, and pH optimums on the y-axis. So the proper temperature and pH needs to be applied in order for these reactions to occur successfully. After a successful mashing, the wort is then drained from this sludge of grain and water, and then it's boiled in order to pasteurize that beer or that wort, and then it is then transferred to a clean vessel, and then yeast is added in order to allow it to ferment. So here we can see a lot of components of microbiology play out, from the use of enzymes in order to break down carbohydrates and catabolism, to the pasteurization of the wort and the fermentation of the wort to produce the final product. Now, it doesn't just stop there. Cells need a way in order to control the flow of metabolism. So they don't necessarily want to break down all of their glucose if they don't have to in order to make ATP. If they're full on energy, they might want to store that glucose to be used for later. And this applies to any metabolic pathway within the cell. There are a variety of ways that this can work. One is called competitive inhibition. So competitive inhibition is, if we imagine this enzyme here, and it's tailor-made for this substrate, well, what happens is if you manipulate what's called the active site, the active site is where that substrate binds to specifically. If you have a compound that's made to bind to the active site will no longer be available for the substrate, and so a reaction doesn't occur. And another method is to have a non-competitive inhibition. And it's non-competitive because it doesn't compete for the same active site. What it does is it reacts with a allosteric site, so a site that is different from the active site. But in doing so, it changes the shape of the active site. So here we see this, convex or concave shape to this enzyme, and once this non-competitive inhibitor binds to the allosteric site, that goes away, and so this tailor-made site can no longer accept the substrate. And this is very important because these inhibitors are molecules that can impede an enzyme's ability to carry out chemical reactions, and it allows the cell to govern what sort of chemical reactions take place instead of it just being a big chaotic mass of chemical reactions occurring. So this is how it plays out in the cell. You might have a reactant that starts here at the top, and this reactant forms a new intermediate compound through a reaction with enzyme 1. And this can occur again in enzyme 2 in order to form a second intermediary compound here. And then enzyme 3 in this metabolic pathway finally gets us to producing our end product, our desired product. However, we don't want an infinite amount of every product. We want the right ratios of every product in order to have an optimally functioning cell. So suppose this product builds up. This product might be capable of doing what's called a feedback inhibition. So feedback inhibition stops the cell from making more of a compound than is needed. So as these products build up in the cell, maybe they're more likely to interact with an allosteric site in one of these previous enzymes. And so say enzyme 1 has an allosteric site, and this end product reacts to that allosteric site, changes the active site of the enzyme. You will no longer get that intermediate step after enzyme 1. Therefore, it will put a halt to this process. Now, after these end products start to decrease in concentration in the cell, then it will kind of let up off of the allosteric feedback inhibition on enzyme 1. And so you kind of have this pumping the brakes process, which is just built into the cell, and it doesn't require any special intervention by anyone with any sort of consciousness. This is just a auto-regulatory process.
Checkpoint 4. Many antibiotics act as inhibitors. What type of inhibition is seen in this animation?
Now, every organism needs two things: a carbon source and an energy source. Because they are organic molecules, all organic molecules must have carbon and hydrogen together. So we can make classifications based upon the carbon source requirements for an organism. An autotroph can consume an inorganic source of carbon. Inorganic sources of carbon are carbon that do not have hydrogen attached to them. So this CO2, this carbon dioxide, is considered inorganic. Heterotrophs can only consume organic sources of carbon. So they have to have carbon that's already been combined with hydrogen in order for them to gain their carbon to do their cellular processes. We can also classify an organism on their source of energy. Phototrophs use the sun as their energy source, and chemotrophs use chemicals as their energy source. Scientists combine those classifications in order to make a compound word that describes both of them. The first part of the word describes where they get their energy source from. The second part of the word describes where their carbon source is from. So naturally, photoautotrophs receive their energy source from the sun and their carbon source from inorganic material. So these would be photosynthetic organisms. Photoheterotrophs get their energy source from the sun and their carbon source from organic material, and some bacteria can do this. Chemoautotrophs get their energy source from chemicals and their carbon source from inorganic sources, and some bacteria can do this as well. And then we have chemoheterotrophs. Chemoheterotrophy is probably the easiest way to get energy and carbon, and so it is the way that animals do it, and most bacteria and all fungi can do this as well. a little side note here, auto means self. So what that means when you see autotroph means that they can get their carbon themselves. So they don't need something else to make organic carbon for them. They can just go get the inorganic carbon. Hetero means other. So heterotrophs have to get their carbon from someone else who's produced it. So because all animals and fungi are heterotrophs, they actually get their organic carbon from somebody else. So in other words, a photosynthetic organism, an autotroph, fixes inorganic carbon into organic carbon, and we can therefore eat those plants or those photosynthetic organisms, or some intermediary organism might, like a cow, and then we can then digest that organic matter later. So we are heterotrophs in that regard. And a little tip for remembering which part of that compound word belongs to energy and which part belongs to carbon is if you just remember a photoautotroph, it becomes quite obvious, because photo has nothing to do with carbon, it's energy, photons. So you'll always know that energy is the first part of the compound word, and then carbon source is the next part. It's a little bit more confusing when you look at a word like chemoheterotroph, and that's hard to decide because chemicals can have carbon in them, and hetero is where the carbon is as well. So if you just remember photoautotrophs is sun, inorganic, then you're gold. Here's our last checkpoint, checkpoint 5. Which metabolic classification are animals capable of? Photoautotrophy, chemoheterotrophy, photoheterotrophy, or chemoautotrophy? Please select one. And that's all I have for today. Thank you very much. I'll see you for Chapter 8, Part 2 next time. Bye-bye.